everybody. My name is Laura Baumgartner, and I'm glad to be the chair of this session. It's my pleasure to announce Professor Ioannis Xenarios as the next speaker. Ioannis Xenarios got his PhD from the University of Lausanne and the Ludwig Institute of Cancer Research. After several years of research at the University of California and leading positions in the Pharmaceutical Research Institute Merck-Serono and the Swiss Institute of Bioinformatics, he got his current position as a professor of computational biology and bioinformatics at the University of Lausanne. Furthermore, he is chief data analyst at the 2030 Genome Center. Ioannis Xenarios' research experience includes a broad range of the field of computational biology and bioinformatics. For example, tackling complex cellular networks, developing methods in non-invasive prenatal diagnostics, early biomarker discovery in diabetes, systems immunology and cancer biology, integrative biology and proteogenomics approaches, and much more. Today, he will talk about knowledge-based network modeling of complex molecular systems in diseases. So thank you very much, Ioannis, for being here, and the stage is all yours.
Thank you, Laura, for the nice introduction. Perhaps what you heard from Laura just right now is that I have probably, and it's actually diagnosed now, an attention disorder syndrome. So I have an attention disorder syndrome. That means any models or any things I'm interested in, I'd love to participate into it, which is never a good thing when you're a professor at a university, because people like to have you focusing on one thing particularly and not to spread too thin. The advantage of computational biology is that you can actually go in different type of domains and support this. So my presentation today will be trying to cover some of the published work that we have been doing over the years. And I was very happy to see that a paper that we published some years ago, more than 14 years ago, is now used by Jerome, and he was actually using that productively beyond the paper, for which we received quite a lot of flack from the community, from both the ODE community as well as the Boolean logic community. And I tend to be very Swiss into this, so I tend to be very neutral. As long as the models can help us, that's what matters. So what I'm going to try to go through is first a discussion I had with the late Marvin Kassman, who is the director of the National Institute for Genomic Medicine. Marv just passed away last August 2020. And we were discussing systems biology and modeling as an avenue for the more molecular or cellular data, and how much he was, as a funder, was facing quite a lot of challenge from people saying it's not the right time to do it. And Marvin had a slide, which I'm always using now, where he actually took the example of what the New York Times was writing on the 9th of October, 1903, where basically the statement was the flying machine, which will really fly, might be evolved by the combined and continuous effort of mathematicians, mechanicians in 1 to several million years. And on the same time, Orville Wright, on the same day, was building his airplane. I mean, it was not an airplane that was doing a transatlantic flight, but it was actually something that made him fly just above 3 meters above the ground, which is already great. My claim is that some of the models that I'm going to show you are probably the same as Orville Wright. They are not as detailed as we wish, but they can actually be useful for the type of purpose we are doing. Now, the goals of models, and for this community, it's much easier, but I always took a course that we gave at the EBI back in 2013, where there are 3 types of models, if we think about this. There are the ones that describe a system, the shape, say, of a shell, which has a beautiful mathematical equation that explains the shell structure. The explanation, which tries to provide with, and this is the Huxley model for axon firing, where you can actually really look at the action potential of a system and try to explain how it works. And you could see that there is about 50 years between these two description of the word that we observe, the explanation of the word, how it works, and the prediction turnaround 2000. That was the emergence of what is called now synthetic biology, where the idea is to construct in silico, in this case, a repressilator. So that's an oscillator that is full of negative feedback loops, into then testing it experimentally and see how it goes. So obviously the last one is the one that we are the most interested in, and the predictions are sometimes easy to do, yet providing that they are right and proving that they are right is the hard part. And I'm going to show you one example where we close the loop. And I must say this is the only one where we really literally close the loop from the prediction to the validation and to the impact into the clinical settings. As compared to everybody of you that have been mainly working on the biophysics or the biomechanical stuff, this one will be very cellular, my talk, and very focused on cancer, which is not really the topics of ours. So I hope you're going to get at least a feeling of how we approach this. Now, there is a slew of modeling techniques that exist, from very large models that describe topologically how each protein could work together. Those are the string that actually Jérôme mentioned in his talk earlier before me, that are most of the time undirected. So we don't know how the flow of information goes in there, who regulates who inhibits who. And those are really what we would call the purely qualitative network topology-based analysis of how a cell works. On the other side, you have something that is more close to most of you, which are ODEs or PDE based, where you have master equation and the idea behind trying to measure each and every of the parameters that exist there. And I must say, in the last 15 years, it's been better and better with the advance in a lot of technologies that both do imaging at the molecular level or actually measure the expression at the single cell level of all genes. The last one was a paper just this week on yeast, where 5,000 genes have been followed during a full cell cycle of yeast, which is probably much the biggest and most beautiful data set for modelers that wants to do ODE-based models. The one I'm going to tackle is something that has been close to my heart for two reasons. The first one is I'm a trained biologist, and the vast majority of the biologists are not really trained and mathematically inclined. There is something also where we tend to think in biology into on-off states, in active inactives, rather than in this gradient, more continuous manner that we have in ODEs. They are easier to manipulate, they are easier to use, and somehow the things that we have in our assays are mainly stable phenotype. We look for a complete differentiation or a complete change of phenotype. This is actually most of the experiments in biology, in cell biology, in immunology, in that manner, for example, are looking at differentiation between a given state and another. And those are really useful in that sense as models. And really a good framework for doing this. Now, there are actually two types of ways people are tackling this modeling. The first one is the top-down. So people that are taking large-scale data, be it anything that you could sequence and get your hands on, from RNA-seq to ChIP-seq, etc., to protein imaging, to high-content screening, to which people try to infer, reduce the complexity, and try to assess the dynamics. There is the other part of people trying to build from the literature what people have been identifying in cells, so the kind of traditional biochemistry or cell biology, trying to construct this and compose the network, try to build up that complexity. So on one side, you try to reduce it, on the other, you try to build it up, so making it more and more complex. Both of them are correct. At the end of the day, you might find a middle ground where both you try to describe how it goes and at the same time try to predict how the system will behave. Now, I have a disclaimer because I always joke about this, but the models that I'm doing are wrong, biased, incomplete, and simplified. And they are a caricature of what we think suppose happens in cells. And ultimately, this is something I've seen because I'm not part of the community of people doing modeling for a living. I'm not in love with my models. I'm ready to throw them away and restart from scratch. And I think this is actually something I've realized over the years, over the last pretty much 15 years I've been playing around with modeling, being not part of that community, is that people become more and more in love with their models and tries to absolutely make it fit to their data, which is a bit of a problem to me. So in that sense, I'm putting the disclaimer for front that all the things I'm saying here will have to be taken with a grain of salt. And I hope we are right. I think we have identified something, but by far, the model does not characterize completely what we are doing in our case. Now, the way one build up models is by taking things from the literature. So this is actually the bottom-up approach. So if you imagine this is the T-helper model that Jérôme had in his presentation. So the T-helper model is a very simplistic model that let's assume this is the truth. Now in the literature, when people do experiments, what they tend to do is several types of experiments. You identify a mutation in a gene that perturbs the system and you look at different genes. You add a chemical compounds that intervene, say, on a kinase, and that has an effect on a lot of genes. Or you add a ligand, an antibody, or others that intervene on this. So when people were doing, for example, a stimulation with a cytokine, say the IL-12, you will see that the interferon gamma, the SOX1, and Tbet will be increasing. So by definition, you're going to draw an edge that says, by adding the IL-12, I have a positive effect on the interferon gamma. You don't know how many steps there was until that one, and you don't exactly how the underlying feedback loops are there. And you could repeat that with a lot of others, say the IL-10 and others. And at the end of the day, the underlying network that is there is confounded or obstructed by a lot of edges that are not direct, and trying to be able to disentangle this. And I'll show you in two projects how we try to disentangle that, to try to be as close as possible to the reality. But it has two assumptions. The first one is that you need to have these nodes into the models. That means you can't model something you haven't put in your model. And the second thing is you should not have a model that is too connected, because everything then is connected to everything. So in that sense, the flow of information that you're trying to capture will be disrupted by a lot of edges that are not there normally. Now, the good thing with a model like this, and here is the model that we're going to use, it has three things that are making up that model. The first one is the node. Each and every of the node is an entity. It could be a protein, it could be an RNA. As compared to an ODE-based, we are not in this case knowing whether we model or we don't assume that we are modeling the protein, the phosphorylation of the protein, or the level of RNA. So this is one of the level of abstraction that we have to do when we build up the models. But the nodes can only take two functions, a 0 and a 1. There is also the edges that could be of two nature, an inhibition and an activation. By doing this, you actually impose on the system that a given node, say the SOX1, for example, inhibits the function of the IL-4 receptor. In that case, you draw a negative edge. Now, with this model and with this set of feedback loops, and you could see some feedback loops that goes, for example, through that path here, you will have a negative feedback loop. But you also have, for example, through that path, a positive feedback loop. It all involves the interferon gamma, which is one of the drivers of what is called polarization of CD4 T cells. But it's actually coming both together on the interferon gamma. So the cell we have to decide where to go. And these feedback loops will tell us which type of function will be present. So the attractors, which is the way we compute on this model, what is the state that the system can adopt, for this particular model, we have only three. The first one is the most obvious one, everything is at 0. There is one where all the nodes that are in red from this model that are actually activated. An attractor is a position in which the system will not budge unless you perturb it externally, either by adding a perturbation or by removing an edge. So that means you have to modify either the structure, the dynamical structure, or the nodes. The last one is a Th2. So this very simple model have 3 states. These 3 states recapitulate, in our case here, the 3 differentiation process that the T helper have, these CD4 T cells. A Th1, which is an interferon gamma led type of response, a Th2, an IL4 led response. So these very simple, if not oversimplistic models, tell us a bit about the biology behind. Now, this work has been then followed up by the group of Denis Thieffry at the ENS in Paris into extending that to categorize more type of T cells, because immunologists tend to love and give names to their T cells. So there are actually quite a lot of other models that actually extend on that basis and have extended on that work since 2016. Now, how do we compute steady states? And this is a even more simplified model, which has only 3 nodes. This we actually borrow completely from the microprocessor design. So a lot of the work I've been doing in the last years was mainly thanks to meeting people in electrical engineering that were doing either check on microprocessor for faults, or we're doing exactly Boolean logic assessment of microprocessors and trying to find their steady states. And in our case, their steady states for microprocessor were the same that we were interested in biology. We had to twist a bit the methods, but all this is based on what is called reduced ordered binary decision diagram, which are a way computationally to compute rapidly the steady states. So let me just walk you through this and try to show you a bit the underlying ideas behind. These 3 nodes are connected with positive and negative edges. And if you actually take all the possible states this system will have, you will have what is called a state transition diagram. The state transition diagram is from a given state, you can either go to one or the others. For example, this is all active. You can either go to this one or go to this one. With 3 nodes, it's actually rather easy to compute the steady states, and you will directly see that there are 2 very special nodes that are very likely steady states. But this is not obvious when you have much more complex, and the more you add nodes and edges, the more the complexity increases. 2 to the power of n is pretty much the number of steady states you have to compute, so it becomes quite complicated. The way people in electrical engineering have been solving this was to do the following. Let's take one state, and they most of the time use the 0, 0, because that's kind of the null state that is easy to catch. And then work through the state transition diagram and say what is forward reachable into that models. So this is actually what would be feasible and reachable. All the others, you have to go backwards in order to go from that state to that state. Then you compute all the backward reachable state. And there is a theorem for this, that if the forward reachable state is not included in the backward reachable state, they are not steady states. So you could, in 2-step, remove them and identify the steady states. So this is the trick, the computational trick that we could use in order to compute the steady states of a system. So now that we have a method that allows to do that systematically on models that contains hundreds, if not thousands of nodes and edges, the question is, can we apply and what kind of models could we apply to this? This was applied to a project that we did with Roche some years ago on modeling the macrophage transition. So macrophages are a type of cells that are important in inflammation, and understanding how they work and how they decide to become a subtype called macrophage 1, 2. As I said, immunologists love to give names to cells, although it's a continuous probably. Trying to be able to do this systematically and try to address this is really one of the challenge that we were trying to tackle. Plus, we had experimental data we could play around. One of the things I was a bit frustrated with the T helper models was that we were imposing the network topology on it. And there might be actually several models that could explain this. So we were not really exploring all possible models given a given knowledge that could explain the data. So rather than trying to apply the same approach as we did for the T-cell, I decided to hire a physicist by training that was interested in genetic algorithms, maximum entropy calculation, and all these kind of things that is not really my bread and butter, to try to tackle that challenge. So Julien Dorier created actually an algorithm called Optimus Qual to actually perform that task. But how did we do this? Well, we did that through a rather standard, if you think about this, for people doing modeling approach. The first was to take a set of knowledge of who regulates who activates who inhibits who. And that we collected over time with people from the Swiss-Prot group in Geneva, curators that knows the field of macrophages and immunology. We had a training set, and what we try to do is to do an optimization, trying to take stuff from the prior knowledge network, take the stuff from the training set, and trying to do a few in silico prediction. And this in silico could be coming from the training or coming from a test set. So the whole idea was reading a training and testing stuff, and trying to identify what was the best model at the end. So the model that we've been playing around is something that is not easily visualizable. It looks like a hairball here, of about 550 entities, nodes, and about 1,800 interactions. So all these are things that contribute to the decision of the macrophages. Now, one of the questions we had here was, if I take this model, can I explain the data? And is there only one model that will explain that? And you could guess that there is not a single model that will explain the data. But there are commonalities among them, and there are certain type of nodes that are more prevalent or more present in order to explain the experimental data. Now, what is the experimental data that we use? These were data generated by the people in Roche that had gene expression, so RNA. In that case, it was even microarray, so imagine that's a long time ago. Flow cytometry, which is looking at the surface, used antibodies at the single cell level resolution. And we had a list of essential genes. So all these guys are things that are considered like the chemokine, some of the surface markers, some of the cytokines that are important for deciding where this monocyte or macrophage goes. And those are all the classification that we have, from monocyte to M1, etc. Those are the perturbation, those are the white stuff that push the system into one place or another. We also had at the same time gene expression data across the landscape with the same type of treatment. So basically, this is our training set that we're going to use our model against and try to see how the models predict or actually is able to recapitulate what we observe. Now, the way to do this, and this is published a few years back, already 5 years ago, time passed way too fast, was to subset the prior knowledge network to try to compute how well the network can reproduce the experiment in the training. So you don't need to take everything, you take a subset and you see how good you are. And you do hundreds of models, if not thousands of models. This is actually quite computationally demanding, actually, like a bit molecular dynamics, because it's actually a process that you iterate over. You try to remove edge, add edge from the literature. You don't create new edge that doesn't exist, right? You take that from the prior knowledge network. You randomly swap the edges between two networks, and you continue like that. And you measure this scoring, which is kind of a fitting function or an objective function against it, until you reach what you hope is the best network. In our experiments here, we run 10 different runs with hundreds of models on 6 independent optimizations. We didn't want to fall into the pitfall of not having a blanket search. The scores was actually replicating about 95% of the training set, which was good. And on average, and this is actually something that is important, we had 500 nodes and a good 1,000 of edges. Well, actually, we could explain the data with only 75 nodes and 113 edges. So this is actually something that is a bit puzzling, because sometimes we wish to put a lot of things in our models, whereas actually it's a bit of a no cameras, or trying to identify a subset is sufficient to understand how it works, or actually to replicate what is in the training set. So when we did that, we merged those one into a model that was what we consider to be the best. And I'm going to go like chasse sur les braises, as we say in French. So I will not detail to that because that would be probably a half an hour talk on how to do this. Just believe me that we get the best network in that sense. And from there, we try to see what does the model, the best network, predict, and how it fits to the data that we are seeing in the experiments. So this is actually, for example, for the wild type. So those are monocytes that are not treated. The prediction is the bottom line, and depending on the gray, on whether it's light or in the I'm missing the word here. Depending on the shades of red and blue, it's also how much we are trustful in our prediction or in the measurement that was done. But what you could see is there is quite a lot of agreement when we have both the expected and the prediction. And any of the nodes that we have in these models that is not measured are prediction on our side. So those are the ones you might be interested to follow. And those are the ones that, in this case, Roche were interested to test because there were some hallmark genes like these guys that were actually in an area of interest that was predicted to be similar in the wild type as these two phenotypes. So the prediction in that sense was guiding the experimental validation or assessment. Because in this case, trying to disentangle each and every of these subsets with a treatment is something that was of interest to Roche. So what I'd like to conclude here is mainly to say, in this case, our aim was really to try to model an ensemble and trying to do that qualitatively on large models, thousands of edges, etc., and have methods for doing this. What we are applying this now is really trying to assess this with a single cell, because the single cell currently tells us with what is called RNA velocity where the cells are going when you are looking at a large population, but we don't know the underlying mechanism behind, and that we are applying this to this. Now this one is good, but not a complete full circle. We have a model, we test it, we train it, we predict, but the validation was not really to the level we wished. Okay, well, if you can't do this, just take another one. That's the advantage of the computational people. You can actually collaborate with different people. So this one was more of a full circle that went from a design point of view to a prediction, to an assessment of the prediction, and back into the patient. So this is trying to identify a combination of therapy for a type of monocytes. And there is a theme here. I love monocytes because they are very plastic, they are complicated to model, and they are still kind of a terra incognita in biology or in immunology and in cancer biology as well. The pro-angiogenic monocytes are monocytes that are present in breast cancer patients, for example, in the blood, in an increased manner compared to the normal population. And the fact that they are pro-angiogenics means that they will induce the creation of vessels inside either the tumor or in different places, but basically to get access to the tumor, to more nutriments, energy, and oxygen. So one of the ideas here was really trying to manipulate this pro-angiogenic monocyte, because we had the advantage of getting the sample from the patients, not use cell lines, trying to manipulate them and try to see how that goes. Obviously, we can't get these samples from tumors every day, but pregnant women that deliver babies, the umbilical cord is full of monocytes. There is tons of monocytes there. So we can actually take the advantage of these umbilical cords to obtain enough material to do that in vitro and then go on the tumor. Now, this has been published. It's already kind of an old story, but this is probably the only one where we really closed the cycle. And it was quite a very complicated paper to publish also, because it has both clinical modeling and experimental in vitro and mouse work, which we published in PLOS Computational Biology. So it's more interesting to publish something like that in a computational biology paper. Now, one of the challenges we wanted to address was, can we, with the molecules that we have down here, identify combinations that would allow us to move two markers that is at the surface called Tie2 and VEGFR from being highly expressed, called high-high, to lowly expressed, low-low. So we binarize already things. We say either high or low. The intermediate, we don't know how to model that. Now, I guess if you don't know the paper and you haven't read that, you can't predict out of this list what would be the combination. So the model has been used as a guiding principle to test and predict what would be the easiest stuff. And it could be 1, 2, 3, 4, X number of combination in order to do this. We decided to go with 3 because that's a bit manageable at the lab, because more than 3 combination, 3 ligands together or 3 inhibitors might be complicated. So the question is, with these 18 factors, how can I move this Tie2 monocyte inside the tumor to a blood tumor? And the reverse, can I put the blood tumor, and what are the factors that would drive them to go into the tumor? So it sounds quite easy, but you first needed to set up the assays. So the whole assay was taking the umbilical cord, purifying the monocytes, which were naive, so not proteogenic, treating them with a set of cocktails. And when we were looking at them, trying to inject them into the eye of a mouse, the mouse does not have any vessels in the eye. This is what you see from something that we took, in this case, from a patient. So it's purified monocytes that are in the tumor, inject that into the eye of the mouse, and you could see that there are vessels that are grown. This is the same patient, but in the blood, purifying those monocytes, and you could see that there is also a bit of that. So this was our assay to know whether we had pro-angiogenic monocyte. So the whole question there was, how many experiments do we have to do? So if we do the exhaustive 1 and 18, that's quite a lot. Obviously, not really an ideal. We ended up doing about 12 experiments, tested the experiments at the end, and we chose a few of them, also with a bit of biological insight into immunology. Technologically, what are the data that was used to model the system? Well, we didn't use single-cell RNA-seq because it didn't exist. We didn't have RNA-seq. It existed, but it was too expensive. We actually used old techniques at the protein level, but we did 2 tricks. The first one was to do flow cytometry. So looking at the surface of what are the markers like these guys at the surface whenever we add either a single ligand or 2 ligands. And you could see that we were not exhaustive in doing all of them. So whenever there was an increase, those would be in red. Whenever there was a decrease, that would be in green. We have done the same in the milieu in which the cells were. We measure cytokines. And the second trick was to take some of the markers that were used as driver, some of the perturbations, was also things that we were, measuring inside our milieu. So we could actually connect the two. Quite a lot of body of modeling is separating these and not trying to interplay the perturbance and the readouts together. And also, what was completely different, because you have to imagine, this has been done on human cells. So every patient's data was important. So we didn't want to waste anything. So it was also kind of a challenge there. Now, to go from the left panel to the right panel, the way we were doing this was to take any of the nodes and say, if I have, say, TNF-alpha and TGF-beta, together they're going to increase the IL-12. They're going to potentiate the IL-12. And you do this for all the ones that we have. So this is actually a rather intricate models, where with these little dots are actually what is called an AND gate. You have to have that and that. And the fundamental question was to say, can we use this and use it to test which would be the minimal set of perturbation that would move this VGFR type 2 high to a VGFR type 2 low. For this, we use again some of the what has been invented in electrical engineering, and that Ron Shamir and Ron Samaga and ourselves have been publishing some years ago, what is called minimal intervention sets that were used for processors that were faulty in the production chain. So the whole idea here is to say, and this is a very simple one, I'd like to know what would be the minimal intervention set if the gene T needs to be activated. And there are 6 minimal intervention sets, of which some, you might say, I'm overexpressing A. It has a lot of effects on others. So those are overexpression or off targets. And it has a knockdown of others. So you need to be able to identify the best. So this algorithm were allowing us to select the ones that were the minimally perturbating and the ones that were driving the push. So with this technology in mind and usage and the model that I showed you before, what we went was to predict what are the possible outcome to move a cell from being a tumor. So that means trying to think therapeutically in this case, I'd like to remove the tumor monocytes and try to push them back to a normal phenotype. And what would be the set of prediction that would move a blood type 2 expressing monocyte into a tumor one. To our surprise, and this is the bottom panel, the prediction that came out of the models was if you type TNF, if you add TNF alpha, PLGF, and H2, you're going to drive completely the blood to tumor. And you could ask a lot of immunologists, they would, out of a million years, not even think about doing that experiment. Since I tell you the story and it's published, obviously it's been proven right. The others part, however, was more complicated. We had 3 classes of predictions, and some of them were virtually impossible to do, like inhibiting the VGFR1. You have to think that we can't do that. We could do that on cell lines. You could do dominant negative stuff, right? But it's virtually impossible to do that on primary cells, either of tumors, monocyte, or onto the umbilical cord-derived monocytes. But there is one that was kind of funny, because there were drugs for the type 2 receptor called type 2 inhibitors. But they were failing. They had no effects in all disease, and definitely not in cancer. So it was a failed drug with no effect. So one of the questions was to say, well, let's try the type 2 inhibitors alone and take some of the predictions and test it. So that's what we did with our modeling and assessment. And this is actually a quite busy slide, so let me walk you through here. On the top left panel, we took the monocytes and we added our combo, PLGF, H2, and TNF-alpha. So we measured the expression by flow cytometry of the 2 receptors, the VGFR and the type 2. This is the white and the black bar. When we did just the 2 that people were thinking it's the right one, we could only rescue type 2 or increase type 2, but not VGFR. When we put the 3 together, it was so high that basically we had to set the threshold as saying, this is the maximum, this is 100%. So anything that's going to be below this will be something that we're going to modulate back. So in that sense, and this is the assay below it, which is UVEC cells for which you look at sprouting and angiogenesis. This is an in vitro assay. You could see that basically you can actually generate angiogenic effects. Around 50% is increased. But when you add these 3, you go up to the 100%. This is what we use as normalization. So first prediction validated, in vitro at least. The second one is we took 3 predictions that were feasible experimentally, and we took the type 2 inhibitors, that is the failed drug. So the failed drug was unable to reduce angiogenesis, was unable to reduce expression of type 2 and VGFR. Out of the 3 predictions, this one was unable to do that as well. I would have been very stressed, to be honest, if all of them were predicting what we were supposed, because honestly, that means we would have a model that is very simple and predict everything. That sounds very fishy. So at least one of our prediction in this case, and there are others that fail. But the ones which was the one which had our favor, there were these two. This one have a strong reduction of expression of TI2 in VGFR. So what we were able to do with this was to close the loop and do this. Now, what happens when you do that on patients? So in this case, you extract the tumor, you extract the monocyte from the tumor, and you treat it either with a combo that reduce the vascularization, and you could see that it's actually reducing quite a lot, or you take the TEMs that are in the blood and you give that combo and you see that it's also increasing as well. We are observing that it's not as strong for the blood TEM to become tumor TEM. So there are other factors that are certainly playing a role here. But from a modeling point of view, a simulation, an assessment, and a validation, this is a closed loop. So in conclusion for this, what we've been playing around was to construct a model out of very not high throughput data, flow cytometry with a few factors, and cytokine bead arrays that looks at cytokine expression or presence in the milieu. Rather than doing the 200 and something combination, we went only for 12. We challenged some of them experimentally. We validated 5 in vitro and 2 in patient, actually. So that's actually quite good, because at least that gave us a bit of a sense of accomplishments. But we have also to be quite honest, we got very lucky with this one. There is plenty of other examples I'm not presenting here where we failed lamentably. And we failed lamentably, not because we were not applying the same type of recipe, because we were actually in things like homeostatic process, which is much more difficult to model with what we have here. So in conclusions, this models has been quite accurately capturing the experimental TEM, was also highlighting some of the crosstalk, which I didn't describe here. It's more in the paper that is described. The model is predictive, that is good. About 80% of the stuff were validated, but that's actually a bit cheating because we haven't been tested more, and see also that they were failed. So at least on the 8 that we have, it's actually good. The TEM are really plastic, so they are actually a very good target. So one of the idea was to go and locally inject those combo inside the tumor to see if the shrinking was done. And this is actually done by people in the Ludwig Institute for Cancer Research, where I am, and that's hospital, and in different companies nowadays, because we reactivated a compound that was a failed drug. With the idea of injecting this is actually for the future. Now, all this work has been done by Marie-Agnès Doucet, who is now actually the lead oncology lead for ICNOS Biotherapeutics, which is actually tackling macrophage and monocytes, and several other people from the Cancer du Sein, the Cancer Center, and people that were more on the computational one. Nicolas Biguet, that's the guy that has the helmet, who's doing the systems modeling with Isaac Crespo, who is a Spanish postdoc that is now working with George Kukos at the Ludwig. Now, I'd like to finish on one of the last words that I got from George Ebox, which is all the models are wrong, but some are useful. And I think this is what I always try to be quite honest about, is that I showed you in the presentation, we only show the things that works very well. I showed you the things that work quite well. And in that sense, these models have been useful, but they are definitely not representing what is happening in life. And with that, I thank you for your attention and welcome any question if you have.
Thank you very much, Ioannis. It was a great talk. It was really, really interesting, especially I think for all of us who are working on this level, on this like low scale level. Are there any questions for Ioannis?
45 minutes, right?
Nothing in the chat.
Seems not.
Don't see any hands. Hopefully this pops up in the first page.
Oh.
Well, in the meanwhile, I will start. there is one question, of course, for many people who are working with models, who are usually dealing with finite element modeling, which where the topic is especially the calculation time. Mm-hmm. From my experience, personal experiences is quite different in the lower scale, so we are much faster. So my first question would be, how long does it take to calculate those huge network models?
So the one that was that we did with the Optimus Qual, we actually have even the benchmarking of this. That was taking about 128 cores for about a month for models that entails roughly about 50 to 100 nodes and about 200 edges. So it's actually quite long. So it's not small. So that's why at that stage, we had access to an HPC. So that was rather easy because we could run at the same time hundreds of models and pretty much consume the CPU cycles. So that means it's not something for the ensemble modeling that you could do on your laptop. Mm-hmm. The other one is much, much more easy, much more fast, because it runs in seconds. I mean, the prediction, literally, I had Marie-Agnès sitting to me while running the miss, and she thought it's going to take hours, and pretty much 30 seconds later, I had the miss. But the model was much more constrained, much more smaller, and the number of prediction was not that big either. So, so on that side, when I talk to Denis Thieffry, who is doing modeling for a living for decades, he's always reluctant to use very large computing to solve a model stuff. So we have also to be quite careful with this because there are other things that we introduce when we do large scale modeling that are instability, numerical imprecision that you guys know even more than I do in that sense, because the more time it takes to compute, the more risk you have to get something that you need to reproduce another time. So there is a trade-off there, basically.
Yeah, definitely. Well, before I still have some questions, more applied questions, but there is one question in the chat from Danilo. So Danilo, if you would like to speak up loud.
Oh, yes, I wrote it, but I can speak it. Yes, because it's really interesting, this kind of modeling. I wonder if you want to implement the Boolean network, trying to switch to directed weighted network. And so in this case, I mean, you cannot use correlation coefficients because it's not directed, but you might want to use probability of affecting the other node.
Yes. So we published, actually, I didn't talk about this because it's another arena that got out of my frustration for the Boolean modeling being too rigid. Is that I'd like to introduce stochasticity, the epsilon term that you guys have, the error term that you guys have, was something I always wanted to have, either at the function level, that means at the nodes, which have a probability to flip, an internal probability, as well as on the edge, which is actually close to a weighting that you're alluding to in terms of the edge, of a probability to be active or inactive. So these are stochastic in function and stochastic in nodes. And we published a paper about 6 years ago exactly on that topic. So you might want to read this one because it's exactly in that area. We use it in a project that was looking at the Yamagata factors, which is the one that maintains stem cells into a stem cell state, to try to see how much stochastic noise was necessary in order to have a differentiation or a stable stem cells. So in that sense, that's our attempt to use that. The thing which is funny, we always developed the methods when we were hitting a wall with our previous one. But that's probably as far as we went to the weights on the edge and really trying to identify what biomarkers were present, etc. So that was what we published. We published that around 2017, something like that.
Thanks.
Yeah. You're welcome.
Okay, this actually already covered one of my questions, but let's go with Maria. She as well wrote in the chat. Maria, do you want to speak up loud as well?
Okay, can you hear me?
Yes.
Yeah. Okay, thanks. Thank you for your talk. I really enjoy it. All that you present. Actually, I use your equation from paper from 2006, so it was really interesting knowing everything you do. So my question is about the bottom-up approach when you develop a network from literature. So when I was establishing all the networks and everything, I realized that the more you look, the more interactions you could find in nodes, and finally you get out with a lot of cross-linking, and well, you end up with a really huge network with a lot of edges. So do you know if there's some like a practical way to decide whether to stop looking for more edges and then to find a network which actually has a representative information flow? Do you know if there's something like that?
So
or
let me
how to attach this?
I mean, this is one of the challenge that we have with modeling. So at one stage, you end up with something like this.
Yeah.
Right. So it has a lot of edges. So you have to. So the first answer to your question is that there is no good rule for this. However, since I work between two communities, the one that do biocuration of protein function, I took care of Swiss-Prot for 11 years, and the people that do modeling, they are now coming to merge together. There is an ITN network called Greek-C that is run by Martin Kuipers in Norway, that is trying to merge these two and to try and provide guidelines on how to do modelings and what to encode. There is also, and the model I showed on the macrophage had contradicting evidence. A paper claiming one thing and another paper claiming the inverse, right? So in this case, your modeling will have directly, you will have to develop two models that take into account these two. And you might not even know which one is the right one, because it's all encoded into the feedback loops. So the way we've been doing this was to try to, from the paper, try to say whether it was sure or unsure. This is becoming a decision of the curator or the modeler. Is to say, I'm not completely sure about these interactions. I'm not sure that this lysophosphatidic acid is effectively inducing the IL-6. We were talking about the weight on the network. We could also put the weight on the edges. Now, one thing that has been also important is that some of them are inferred. So we sometimes tend to, and that's why we were not looking too much in the discussion, because that was more hypothesis. It might, could, perhaps, blah, blah. It was more trying to be close to the figures that describe effectively what was going on biochemically. What I'm trying to say is that there was no good rules, and I think you're gonna have to do a judgment call yourselves on how much this information will be useful for your models. And if I look at what Jérôme has presented just in his talk before, making a trade-off between not having all the cytokines, but taking all the cytokines and the MMTs and the metapolymerase will be actually something that is a trade-off to choose between the two. The way we were trying to do that was really to try to classify and structure them. So we did that for pancreatic beta cells, etc. And every time we realized that after a while we had to stop because it was too connected. And then we had to remove the edges from some of them to get to a lower, to something that was usable and understandable as well, because that was also the trade-off we had. So I'm giving a very long answer to something which I'm somehow not having the answer now.
Okay, well, yeah, I understand what's the point of everything. So thank you.
You're welcome.
Okay.
Thank you, Miss. I think if I can jump quickly in that discussion, presents the opportunity. I think one of the decision to take, and it's not really an easy decision in what I have presented, which is actually the work of Maria during her PhD thesis, I say to it now. So we have then this network of only soluble cytokines. So this is relatively reduced compared to what you've presented, and is even further reduced after the genetic algorithm application.
Yeah.
So it's quite handleable. Now, when we validate that network independently, what we find is that, so for one stimulating node, so if we take the mean response of the network for each family of stimuli, we have actually great results.
Mm-hmm.
Now, if you look, so now you have a matrix of 81 points of verification if you start to look for each stimuli, each node.
Mm-hmm.
And here you see that you're concentrating the error on some nodes,
Yes.
on some of the points. So if you make the count, maybe you're concentrating the error. I don't remember exactly, Maria, but it was about 8 to 13 nodes over 81.
Mm-hmm.
So here comes the question: do you accept that, or you don't? If you don't, what we're seeing is that you start to have into transcription factors,
Mm-hmm.
and there is a high risk to lose the control.
Exactly. So, I mean, there is one thing that I completely did not describe, is that I try to stay as far away as transcription factor as possible, but they always bite you back in the model, because they are effectively regulators. We can't say master anymore nowadays, but they are heavy regulators of the decision that the cells will take, or the environments in which the cell will be proliferating, or in your case, an osteoarthritis type of thing. So in that sense, they have to be used with very big caution. And those are the ones for which we have to be also honest. I mean, it's getting better and better by the days, for which we don't have hard evidence of how these factors effectively perform their task. We know that they bind, and in some database, the fact that it binds means that they drive the process. And the binding doesn't mean that it drives the process, it means that it can be there, and it might not be there at all for a long time in that context. So these kind of aspect is hard to model correctly. And it's good that you're mentioning this. Most of the place where we failed, where actually the prediction was not right, was when we had transcription factor being part of that equations. The only place where it works with the transcription factor, where we were modeling ontogeny, or very like the fingers modeling, this is actually fine. The Hox genes, etc. All these kind of things are programmed in such a way that basically you have one step after each other, and you're locked inside a state. There is no way to come back. For the monocyte, for the T cells, which has plasticity in coming back, this is actually virtually something that is much more complicated to tackle. And the transcription factor might be just in a few seconds or minutes and drive the whole chain of events, and then it could come back again like a wave. So that's where we have challenges, I would say, here.
Thanks.
Welcome.
Okay, I actually have a quite related questions to everything that was already asked. I was wondering about, like, time and dose dependencies.
Mm-hmm.
Like, which is really a huge topic as well, I would say, in the scale. I was wondering if you remember to have ever changed, for example, like the effect of the activating or inhibiting node, for example, like IL-1 beta. So you assume a certain amount of pro-inflammatory cytokines, which might be inhibiting, but another amount might be activating. So did you do anything related to this in your models? So changing just activating to inhibiting, just to check how the model would behave when you just change one node activation to an inhibition, or whatever experimental information about those or time dependencies.
So the only example, this is another set. So I had like 150 slides, so I reduced it to that. The only case where we had to do this was in a yeast model. So let me shoot that one. And it's so there's two things. First is Jérôme's paper that he mentioned, where we introduced the sigmoid function, was where we had for the interferon gamma, which has 3 stages or 3 levels, a high, medium, and low. And rather than doing a high, medium, and low, we decided to move towards more ODE based. So that was our first attempt. The problem with that is, besides the Hill function and some of the parameters, it becomes more complicated to say what is the right parameter to use. And you have to think, 2006, a dude that is a biologist plus another one that is a biologist plus a bit mathematics. So not with the proper training in modeling. So we were trying to attempt that. So our second attempt was to rather encode that into the rules of the logical function. So let me now just share again my slide, sorry. And this is on a model which is the septation, so the separation of yeast into two daughter cells. And there is a wall that comes at the intersection between the two. So we did, and there is a lot of players, and it's a cell cycle. So basically it happens again the next time in the next daughter. So you have a lot of proteins that have been studied biochemically. But the thing which we knew was that there was these CDKs, which is a set of kinases that were in different levels. The first model we did, and I'm going to go very fast on this. So there are people like Attila Nagy and others that have done an ODE base, a very simple 4-equation system to try to explain how each and every of the states were regulated. The model we did, which was exactly applying the same approach as we did with what Maria was asking about, where do we stop? And we stop after a while because it's becoming too complicated. When we did this one and we did prediction, this one failed lamentably. Our prediction was saying we're going to have only one wall. And when we did the mutation, poof, we got 1, 2, 3, 4, 5 walls. So clearly, we were doing something wrong, and we were moving away from exactly what you're mentioning, Laura, which is different level of the system will trigger certain type of feedbacks. So in order to solve that, we went back and we said, all right, we need to do that differently. So that's the one so that's the failure, doesn't work. So we took the CDK and decided to say, we're going to place them in 3 levels. An interphase in early mitosis and late mitosis. And we're going to connect them to the model. So as compared to the first model, which was a failed model for that part, this one is much more stringent. So we reduce the complexity, but we also open up a very big can of worm for the modeling, is that we want to have something that is more tunable. A bit like your sensitivity analysis for high, low, medium of a given cytokines or things like that. Or IL-1, for example, being another one. With this one, we could actually do the modeling. We did the assay, and surprise, surprise, my PhD student was very happy. So we did the modeling, tested it. I go very fast here. It had a prediction that was absolutely not something that the biologist had. Paulina did the experiments, and she could graduate and get her PhD. And we published the stuff. So that's the 3 years compressed in 10 slides. This one was our attempt to try to code these 3 levels of interaction. But it's an attempt which is still not answering your question, because here you'd like to have more of a continuous value that you could play, like in a Hill coefficient, or some kind of granularity along this. And this one, we haven't been finding a good way to express that. And also, I must admit that about 4 years ago, I wrote a grant for the Swiss National Fund to hire a mathematician for this, because I could get the feeling of what would be cool to do, but I don't have the tooling to implement or think about this, and also understand the limitation. Because one thing that Jerome has probably seen in the paper that we published in 2005, on the extremity in the low and the high, you have instability that goes above 1, which is very bad for an equation, right? It's not bounded between 0 and 1. And this is something that we coded, what we hard coded into the model, which now retrospectively, I could say that years after, because my professorship is done, so I'm tick box, this is actually bad because it's not completely proper mathematics in that sense. But thinking back in that time, this is not really what we were after. But I mean, all that for reproducible science, I did contribute to a weapons of mass distraction. Apparently, Jerome used it well, so he knows how to use it correctly.
I think we did what we had to do. That we did. So was one master student who is actually instructor in the hands-on now. We did a sensitivity analysis by using by making a sweep of all the possible parameters, and this is where we actually spotted actually this instability. So we know that in all the models that we are away from this unfortunate set of parameter values. So that's fine. But I think also that, and that's take-home message for everyone. Huh? I think that before you start to interpret a model, and obviously before you start to publish, make a sensitivity analysis. You might have surprises.
Oh yes. Yes, and the reviewer didn't ask us this, but if you had said, we would have spotted this in 2012.
Well, we actually already consumed 5 minutes overtime because it was really, really interesting. No, not at all, because it was really interesting, and I could keep on with this because I have a bunch of more questions actually. But for the sake of our lunchtime, I would suggest to stop here. I was quickly checking before, I didn't see any more hands on, so I think we're quite done with questions. I would like you to thank you again, Ioannis. It was really an absolutely great talk and really interesting for all of us working on that level. And yes, so I wish to all of you a really nice lunchtime, and see you in the afternoon again with the hands-on.